Hello, good morning. So this is our first time to study via online. So last time we discussed about, what did we discuss about last time? The DNA replication. Okay, so when you say DNA replication, it means uh, our body or our DNA needs to replicate. Replication from the root word itself, replicate. Why do we need for our body or for our genes or our cells to replicate the DNA? DNA is important, is vital and crucial functions needed by our body. In the DNA, information are stored, so it's important for our body to replicate this stored information. So in order to replicate the stored information, the DNA will make an exact copy of it, especially during cell division. So we all know that in cell division we have the mitosis, which is cell division by the somatic cells, and the sex cells via meiosis. So after the DNA replicates, so there will be two strands of identical DNA. One from the mother at the parent strand and one is the daughter strand, in a manner which is called as a semi-conservative manner. Now, let's go to our transcription and translation. So, from the DNA, from replication, we go now to the RNA, or the making of proteins, and that involves transcription and translation. There are three serious stages during transcription and translation. Number one, this is too thick, I'll change. Number one, the first is the initiation, followed by the elongation, or to elongate, and the third one is the termination. So these three are processes that involves via transcription and translation of RNA to proteins. First, as an overview, we know that genes in the DNA contain information to make proteins. So if you try to look at this, these are the genes, and then it unwinds, and then remember the DNA replication, the helicase will unwind between the what they call this one, the leading and the lagging strand. So that is about DNA replication. But during the RNA replication, the cell makes RNA copies of genes that are needed. So as you see here, the red part in here, that is now the RNA, and it needs the code of the DNA in order for it to have its own code, and then later on will be transcribed into the ribosomes and read as amino acids or proteins. The mRNA is read at the ribosomes in the rough ER. So if you notice part of the cell, like the cell nucleus, here example, remember the cell, we have the cell nucleus. This is the cell nucleus, and the chromosomes are here and the DNAs are here. So outside the cell nucleus are the highways, networks and highways surrounding the cell. And these highways, networks and highways contain tiny bumps in which we call it the rough. So these tiny bumps are known as proteins, which giving it or making it rough. So this is now what we call the rough endoplasmic reticulum. So in this rough endoplasmic reticulum, this is where mRNA is being translated via translation and synthesized into proteins. So in the ER, protein is produced. So how does DNA transfer to mRNA, to tRNA, and then becomes a protein? So we have some key players involved. There are some key players involved in this process. So first, there are three kinds of RNA. From our previous lesson, we know that RNA is a structure the same with DNA, but it doesn't have without oxygen. Okay? I'm sorry, it's not oxygen. Okay, but the structure of RNA, the structure of RNA differs from the DNA from the word itself, deoxyribose, because one carbon of the sugar group doesn't contain oxygen. Okay, that's the definition. So there are three kinds of RNA. RNA stands for ribonucleic acid, so it's a ribose sugar. It has a 5 or a pentose carbon, and it has three kinds. First, we have the mRNA. M, it means messenger RNA. Okay, and the messenger RNA, from the word of messenger, it carries the information from a gene to a DNA. And then the next one, we have the RNA, or the ribosomal RNA. That's why it's called RNA, ribosomes. It carries out enzymes like role. Later on we will discuss. And then the tRNA, which also refers as the transfer, because it will help transfer the RNA or the amino acids during the protein synthesis, or most commonly known as the translation RNA, because it's involved in the translation process during production of amino acids. So if you see this one, these are the parts of these are the different kinds of RNA. So messenger RNA, this one messenger RNA. This messenger RNA is coming from the DNA when it pairs with the DNA strand, the upstream strand. And then the ribosomal RNA, later it will be found in the ribosomes during the translation of proteins. And the tRNA that functions like an enzymes, which helps and brings amino acids and an anticodon as well. So the following here in the mRNA, look, this one, the base, the nitrogenous bases are called the codons. They are found in the mRNA, while the nitrogenous bases found in the tRNA are called anticodons. So a codon is always paired with an anticodon so that they form a base pair. Okay, so let's go now to transcription. So from the word of transcription, it means to transcribe. So from the word itself, transcription, or to transcribe, we see here the following. So we see here the following. So this is the gene 1 and the gene 2, I use red. So this group, or the gene 1 and the gene 2, will be a series of genes that will be transcribed. For example, this part here, later it will transcribe this part here, it will be transcribed, this part here will be transcribed. So if we magnify the following, so usually where the RNA polymerases or the flow of transcription always starts with 5' to 3'. And here 3' to 5'. It's going like that. So the DNA, during the DNA transcription, we have this enzyme called the DNA polymerase to initiate the transcription after the helicase unwinds the bonds, the hydrogen bonds. Here in the transcription, we have the RNA polymerase. The same with the DNA polymerase, the RNA polymerase initiates the